Okay, I think we should get started. Can you hear me in the back? Good. So I'm Leslie Lonwan, the CSO of the BioFrontiers Institute, and I'm just going to give you a little bit of introductory information before introducing our speaker tonight. I also have to warn you that I just flew back from Hong Kong last night late, and so I might be a little... You can't hear? Okay. Ice, can you... So maybe the people in the back could hear me, but not the front. Is that better? Okay for you guys? Okay. That would be mean. Anyway, so many of you know about this series of lectures, and we're really proud of them. We think that they've been a great success, and it seems like just yesterday that we had the fourth one from last semester, and now here we are in September. And we really like to talk about our impact into the community, and I think it's really telling that's interesting. It seems to our anecdotal observations that there are a lot more people from the community here tonight, I think, than some of the ones last year, and I think that's actually a really good thing. So thank you all for coming. So we've got two more this semester, and they are from two of our best people in BioFrontiers. You haven't heard from them yet. Ryan Layer is relatively new. He's an assistant professor in computer science, and he's going to be talking about AI and precision medicine, and he will do his best not to use a whole lot of equations. He and I are making sure that he understands how to speak to a broad audience, and I think you're going to love it. Christy Anseth, many of you know, she is an associate director of BioFrontiers, and she's going to be working, talking to us about regenerative medicine. And she does a variety of things that are afflicting many of us who've needed things like hips replaced or knees replaced. And so we really want her to succeed. I particularly do. So they will be speaking and sign up to attend these. Many of you already have. So I think we're going to have a good turnout for that. So what is BioFrontiers? As many of you know, we are a research institute here at the University of Colorado, and we're working on problems that can affect all of us in both health settings as well as in disease settings. And we've got wonderful interdisciplinary programs that people are collaborating with, and you've been seeing people talking about these various things. And I think you'd all agree that what we work on in this institute is really varied, and it, I think, exemplifies what we started out to do so many years ago. So these are broad general areas that we work in: regenerative medicine, Down syndrome, computational biology, the degenerative diseases, as well as parasites and pathogens. And you've heard about some of those, and you're going to hear more about the degenerative disease tonight. So we support a lot of programs here. The IQ Biology program, as you know, is a PhD program, interdisciplinary quantitative biology, that was started by BioFrontiers about 12 years ago. We also take care of a lot of facilities that allow us to do many of these aspects of the things you're going to hear about scientifically and from an engineering standpoint. We also really support biotechnology. We like to do it homegrown, so people who are here, or from external relationships where we have biotech companies residing in this building. And that's worked out actually extremely well. So we are always thankful for support that we could receive to support existing IQ biology students, as well as how important this is to change people's lives and impact society. So please, if you can support us, do. And this is our current flock of IQ biology students waving to people and greeting them. So, not to take up any more of Roy's time, let's go on with the show. So tonight is Roy Parker. He's the current director of BioFrontiers Institute. He's an investigator of the Howard Hughes Medical Institute. You'll hear a little bit more about that on the next slide. But I think you're going to see that this talk is extremely relevant to today's society and to many of us. He's a distinguished professor in biochemistry. He's a wonderful RNA scientist, and he is connecting RNA biology to different neurodegenerative diseases. He's also a prestigious, he is a prestigious member, but he's a member of the prestigious National Academy of Sciences.
It's that Hong Kong jet lag.
No, you are prestigious. And a wonderful colleague. And so I'll turn this over to Roy.
Thank you, Leslie, and thank you all for coming. It's really great to see so many people, and it's fun to talk to you. I want to start my talk with two quotes, if I can figure out how to, okay, ice. Two quotes from this man. Who am I? Why am I here? And I'll point out, if you know who this individual is, you're in the age demographic that you should be paying attention to neurodegenerative diseases. However, if you actually remember who he is, you're probably in pretty good shape. So for those of you who don't remember who this is Admiral James Stockdale, really a decorated admiral who was Ross Perot's vice presidential candidate in 1992. And these were the first two lines of his vice presidential debate. Okay, but who am I and why am I here tonight? And as Leslie talked about, my career has not been in the area of neurodegeneration. In large part, I worked many years on RNA decay, on other aspects of the biochemistry of RNA molecules in our cells. But I'm right about here, and this is the slope of which at age you start to get neurodegenerative diseases. And so a few years ago, I started thinking, maybe I should pay attention to these, not just for personal reasons, but also because this is a big deal in society. And so my lab has shifted into studying neurodegenerative disease, at least initially from a perspective of RNA biology. What I want to do today is talk about really three areas. I'm going to talk a little bit about the impact these diseases have on all of us and society. I want to really emphasize the biochemistry of these diseases, and in particular, I want to talk about how these diseases spread by something called prions, where one neuron infects another and makes the second downstream neuron sick. And then I want to talk about, what can we do about this? Right now, we can't do much, but we can do some lifestyle things. But also, what might be coming in the next 5 or 10 years and how that might play out. And throughout these parts, I'll weave in some of the work that people in my lab do to try to address these issues. I want to start with a disclaimer. Neurodegenerative diseases are really complicated, and I can't cover everything in tonight's talk. So I'm going to leave out some big chunks, how many different parts of our brain interact to affect these diseases, how inflammation in the brain affects this. Just like inflammation in our knees can affect arthritis, we have the same kind of inflammation in our brain affecting these diseases. And I won't say much about genetics and lifestyle choices, except for at the very end, about how these impact these diseases. But I'm happy to talk about this as we go on. But what I do want to talk about is about the biochemistry of these diseases and how that affects the process. To get there, I have to start by talking about the brain. And I want to remind everybody what the brain is. The brain is a collection of 100 billion neurons, at least for most of us, that make 100 trillion connections between them. And so that creates different circuits in the brain, and those different circuits control how we move, what we remember, and what ideas we might have tomorrow for breakfast. And this is really driven by these properties of neurons, because what neurons are cells that can pass electrical activity from one cell to the other. So you can have a neuron here, which gets triggered, sends an electrical signal down, it jumps across a special region called the synapse into the next neuron. And so in a very simplified form, you can think of these just like wires, and just like you have wires in your house which control your lights, and different wires which control your garage door opener, we have different neuron circuits that control different behaviors and control different ideas and different memories. Neurons are really amazing cells. Each of these dots is a neuron in the brain, and they're really packed close together, and they have lots of connections between them, and they do lots of really tight circuits in the brain. But there are other neurons which are really huge. This is a neuron in all of us, which starts down here in our toe, and when you stub your toe, it goes zzz, and then up here to your brain, you go, ouch, right? So this neuron in some people can be, up to 2 meters long. And that's really amazing, right? So one cell, most of the times we think of cells as really tiny thing, 2 meters, okay. What happens in neurodegenerative diseases is we lose neurons, progressive loss of neuron dysfunction, and then loss of neurons. And these are diseases you're all familiar with. Alzheimer's disease, Parkinson's, but there's a lot more. ALS, Huntington's. I'll talk a little about chronic traumatic encephalopathy. This is CTE, the thing that athletes get from repeated head trauma, okay? And all of these diseases share several properties. The first is they get worse over time. I'm going to use the example of Parkinson's, where you start out with tremors or movement disorders. Over time, which can be months or years, that gets worse. You might lose your balance and fall, and then over more months or years, you have the inability to stand or walk. So progressive loss of function in terms of our abilities. And that reflects progressive loss of neurons in your brain. So that early on, you might have lost a neuron, later on, you've lost 3 in this cartoon, and later on, you've lost 7. So what's happening during that transition from mild symptoms to more severe is your brain is slowly losing more and more neurons. Yeah, this is scary, right? So why do I care? Why should you care? This affects all of us. I don't think I have to tell this audience that, right? The Alzheimer's Association in the United Kingdom estimates that 50% of people will be affected by some form of dementia, which is a broad class of neurodegenerative diseases, either by getting it or caring for someone who has this. I actually think this number's low. I suspect every one of us in this room has been affected either because of a parent, a grandparent, a friend who's either had this disease or cared for an elderly person who had the disease. So I think it affects all of us. It also has a huge impact on society. Alzheimer's, which is about 50% of neurodegenerative diseases in the United States, is the 6th leading cause of death. It costs over $350 billion a year, even in 2020. But I think these things pale to the emotional impact on families and caregivers. If you've ever been involved with someone who has dementia or caring for someone, it's a huge emotional impact, and it's really traumatic for not only the patient, but also for the family. The other reason I got interested in this and decided to refocus my lab on this is it's getting worse, right? So in 1980, only 4% of us were over 75 years old. In 2050, it's estimated that about 13% of us will be over 75. And the number one risk factor, you saw in that first graph, is age. So, this is a great success of our treatment of heart disease and improvements in treating cancer. So that's all good, but now we're all gonna live longer and we're all gonna end up getting neurodegenerative disease, so we have to figure out what to do about it. All right. So what causes the dysfunction and death of neurons? And so to talk about this, I have to back up and remind everybody how all the cells in our bodies work, including our neurons. And so all of our cells have 3 important classes of molecules. They have the DNA, which is really the genome, the repository of information. You can think of this like the blueprint for everything that goes on in our cells. When you want to use a part of the I've lost a, are you still here? Do we have ice solves everything. Okay, so when you want to use a particular gene, you copy it into what's called an mRNA or a messenger RNA. And you can think of this like a temporary copy of the blueprint. And then that copy is then sent to a factory, which is called the ribosome, which makes what's called a protein. And proteins are the machines which do almost all of the work in our bodies. Oh, thank you. Okay, it's not quite as bright. But, okay, so there's lots of proteins, and you're familiar with these proteins, right? Your hair is made up of proteins. But you have proteins in your gut. When you eat food, proteins are what digest that food so you can use the nutrients in it. When you move your arm, the proteins actin and myosin slide past each other to allow you to contract your muscle. And then, one protein everybody's familiar with is insulin, which tells your cells to take up glucose from your blood to use it for energy. And so these are all proteins. And you'll notice these proteins all have shapes. And those shapes are what allow them to do their job. What happens in these diseases is the proteins get the wrong shape, all right? So they're what are called protein folding diseases. So imagine you had a protein which you sat in, okay? And I know this is a little bit of a reach, but if that protein was folded up properly, it would look like a chair and I could sit in it. But if it was a protein that was folded at Tom's house and he got it from IKEA, it might have ended up like this, and it's non-functional, right? So it's not only non-functional, it's worse than that those non-functional proteins then clump up and form protein aggregates which are toxic, all right? And so fundamentally, all these diseases are what are called protein folding diseases. The proteins misfold, they have the wrong shape, and they cause problems. And all of these neurodegenerative diseases that you've heard about, frontal temporal lobe dementia, degeneration, another common, not so common, but prevalent neurodegenerative disease, ALS, Parkinson's, Alzheimer's, all of these are driven by a misfolded protein. And the protein that does that is different in different cases. In ALS, it's mostly TDP43. In Parkinson's, it's a protein called alpha-synuclein. And so different diseases give different misfolded proteins give different diseases. And then if your proteins misfold and neurons die, your brain loses neurons, it shrinks, you lose circuits, you can't remember things, you can't move. And so that's the problem with these diseases. And this is a problem because we essentially do not replace our neurons. So if you lose a neuron, it's gone. And so eventually you lose enough neurons and you're compromised. I want to tell you about a huge unanswered question. All your neurons are dying. We don't know why they die. We know that these protein clumps, these misfolded protein aggregates, drive neuronal dysfunction and death. But nobody knows why they die. And one of the things, this is a huge unanswered question. And so one of the things you're going to see when we talk about therapies is there's nothing targeting what causes the neurons to die because we don't really know. There are 10 different ideas, at least, and they could all be right, or it could be just one of them, and we just don't know the answer. This is a big problem. This is something that we try to work on in my lab, but we haven't solved it either. All right. But why do these diseases spread? You can lose neurons, like you can have a stroke, and you lose a part of your brain, but that's it. You can even recover sometimes from a stroke because your neurons can make new circuits and actually restore function. But these diseases spread, and they spread through a mechanism that I'm going to call a prion mechanism. So imagine we have a whole bunch of proteins up there at the top, and they're all properly folded, like a chair. But this guy in the middle forms the aberrant structure. And I'm going to call this a seed through the talk. And not only is it an aberrant structure, but it's infectious. So this aberrant structure can bounce into one of the normal proteins and turn it into the bad guy. And again, and again. And so you end up with all these guys in the aberrant shape. So it's not just that one protein misfolds. The guy that misfolds can then bind to another one and turn it into the misfolded shape. That's what's called a prion. And this is what you've heard about is mad cow disease and kuru are prion diseases. These are cases where they're misfolded proteins transmitted in our food. So mad cow disease is where you have a misfolded protein that you eat when you have some cow. It gets into your stomach, through your blood, into your brain, interacts with the proteins that are normally there, and turns them into the aberrant shape, causes neuron toxicity, neuronal death, and then you end up in mad cow disease passing away relatively quickly because it's extremely fast. I just find this amazing because I want to emphasize how different this is from other diseases that we think about. You know, viral diseases driven by viruses, bacteria driven by bacteria, parasites like Giardia, a bug gets into you from outside. These are all agents which are living things that are causing the disease. These prion or prion-like diseases are driven by our own proteins misfolding and then triggering other copies of our proteins to misfold. And because they can go from one neuron, you have this neuron that's full of these misfolded proteins, maybe that neuron dies, releases those seeds, and they get taken up by the next neuron, and then that neuron does the same thing. So this is why these diseases spread through your brain. And so if you look at different stages clinically, people have characterized Alzheimer's into different stages of disease where it starts in this kind of a central region of your brain in early stages and then spreads to the forebrain in the middle stages and then eventually spreads throughout your whole brain. So, and this is all because of this prion-like spreading of the disease. All of these diseases spread by this prion mechanism. And while it's not known, I would speculate that any of these neurodegenerative diseases, which are progressive, are going to spread by prion-like diseases, because otherwise, why would they spread? So, all right. I want to say a few words about Alzheimer's, because Alzheimer's, over there on the far right, has two proteins that misfold, A-beta and tau. And I want to spend most of my talk going forward talking about tau for two reasons. One is because I think this is the most important protein in Alzheimer's disease. It's also really important in lots of other diseases. And it's also what my lab works on. So a little bit of selfish focus there. But, all right. So, but Alzheimer's disease, many of you have heard of A-beta or beta amyloid, right? So what's going on there? And so really what happens in Alzheimer's is you have what's called a beta amyloid plaque, or what's called amyloid. And it used to be that people thought this was what caused Alzheimer's, and it certainly contributes. And then, but I think the understanding now is that this amyloid plaque does something, this is outside your cells, to the neurons, to trigger those neurons to start making prions of tau. And then those prion-like taus spread, leading to neuronal dysfunction and death. So you should really care about tau. And so the other thing about tau is that in 80% of neurodegeneration, tau is the protein which drives it. So this is like CTE, Alzheimer's, some other diseases like PSP or CTE. And I should say there are many diseases which all converge upon tau. So this is like the crux, I think. And I just want to mention that this can be even things like virus infection. If you get measles and you're not vaccinated, 1 in 500 to 1 in 1,000 people who get measles and are unvaccinated, 10 years later, develop a lethal tauopathy called subacute sclerosing panencephalitis. And so if you have friends who don't think their kids need to be vaccinated for measles, let them know, okay, they're probably going to be okay, but there's a 1 in 500 chance that they're actually going to pass away 10 years down the road. All right. So we're going to talk about tau. So what is tau? So tau is a protein which helps our neurons work. So remember this long neuron I talked about. So when you have a really long neuron, neurons are kind of like spaghetti. So if you're going to have a neuron that goes like all the way up my spine, they have a structure within them which helps stabilize them. It's called a microtubule. And what tau does is bind to microtubules and help keep it sturdy so that you can have these really long parts of neurons. But then what happens is sometimes, here's our neuron, and these are tau molecules here, these little things. In our cells, nothing is static. So what's going on is these molecules are bouncing around. They come on, they come off, they come on, they come off. Now let's imagine that this neuron got a seed. Now when these proteins bounce off, they might run into the seed and get converted. And so now, over time, much of the tau, if not all, can be converted into this abnormal state, and that's a toxic state, and then the neuron will be dysfunctional and eventually die. So this issue, how does a seed get into a cell and then create new tau aggregation and new tau seeds, is really an important problem. And this is one of the things my lab works on. And we got into this many years ago, not many years ago, a few years ago, because as an RNA biologist, I was always fascinated by this observation that many of these proteins bind RNA. And that includes tau. And generally, people who study tau didn't even think about RNA. And so we got interested in studying this. And the first thing you might ask is, how do you study this disease if it happens in people over decades? Well, we'd like to look in some of the brains of our friends. We can't do that. So we use several different models. One model is we use mice. So we have mice which express a tau which causes disease in people. There are mutations in tau which give you disease. And if you put that into a mouse, it gets disease. So you can use mice. Mice are great. They're the best thing we have to a human brain in the lab setting, but they're very slow. So we can also study this in human cells in a dish. And we can do that because these are prions. They can generate new infections. So we can take a little bit of the brain of a mouse, or people have also done this from patients who passed away and donated their brains. And if you take that extract and you put it into cells which make tau, then it'll start making new tau aggregates. So this because it's a really nice, fast system where we can study how does these molecules grow, how do they make these bigger structures, and what might you do about that? And then the third system we use is post-mortem tissues. If we learn something in the cells, in a dish, or in a mouse, you have to go back to human samples to see if that's really true. And so here we've been very fortunate to work with Stan Prusner at UCSF and Nadine Bakker at the Neurodegenerative Institute in Phoenix. They both have extensive brain banks, and so we can actually go and look in the brain of someone who, when they passed away, they donated their body to research, and ask, is what we see in animal models or cell lines true in humans? And that's super important, because otherwise we could be just studying some random artifact. All right, so a few years ago, this gentleman, Evan Lester, joined my research group. Evan was a PhD student here, but he was a medical student down at Anschutz. And this is a great program where some of the medical students get their MD there, but they also do a PhD up here. And so this is a great marriage of strengths, because he brings a clinical perspective to my research group, and we also then train him in really robust biochemistry. And Evan's now a resident at the Rockefeller in New York. Okay, and so what Evan wanted to do is learn what molecules were in these tau aggregates. What's actually there? And so we might learn from that how they grow and why they're toxic. And so I'm going to show you some pictures of cells. In order to do that, I have to show you what a cell looks like in these kinds of images. And so this is what cells are going to generally look like. So cells, human cells, have a chunk of them, and it's typically going to be blue because we add a dye which glows blue. It has the DNA in it, and it makes the mRNA up there. So remember this, we're going to copy the DNA into RNA. And then that RNA goes out into the cytoplasm where the factories are which make the proteins, okay? So you'll see most of these cells you're going to see have a big blue dot. That's the nucleus where the DNA is, and then a bunch of stuff around it. So this is what Evan learned to reduce his PhD to one slide. He'd be disappointed. But he learned that tau, when you have these tau aggregates in the brains of mice or in dishes, They have lots of RNA in them. That's a story for a different day. But he also learned they had lots of interesting proteins. And this one protein I'm going to talk about is shown in red here, SRM2. You can see in normal cells, that blue nucleus where you're making RNA, that's where the protein is. But if you have tau aggregates here, where we've seeded these cells, so they started making new tau assemblies, you can see the red protein moves from the nucleus out to where tau is, okay? And this red protein makes RNAs. So this right away says, well, if you're losing a protein that makes RNAs, maybe you're messing up what RNAs you make. So the first thing we do is we say, is this true in people? So we go and look in brains of people who've passed away from one of these tauopathies. This is, I'm just going to show you one image from a patient with cortical basal degeneration. It's just another disease driven by tau. That's not me. Okay, so this is the patient. And what you'll see here is there are these green tau fibers. These are these, is that me? Oh, okay. So these are the green tau aggregates, and you'll see this red protein is now found all over them. So normally it's in the nucleus. Up there you can see it in the nucleus. It's left the nucleus, and that's going to change what RNAs you make, and it's now out in the cytoplasm. And this is true not only in this cortical basal degeneration disease, it's true in Alzheimer's patients, it's true in frontal temporal lobe dementia, another disease. All right, so why do we care about this? We care about this for two reasons. One is because this red protein normally makes RNAs, when it's now out here in the tau aggregate, you're not going to make the right mRNAs, and we've shown that's true. And we know that people who have these diseases don't make the right mRNAs. We don't know if those are directly connected yet, but that's something we're working on. The other reason we care about it is that maybe, and I'm going to show you some movies in a second, this can tell us how these tau aggregates grow in cells. Because we'd really like to understand, not just when the seed gets in, how does it become a bigger thing, right? Because if we could understand that, maybe we could change it. How do you figure out how something happened, right? Now, you can have pictures, such as, cells before and after, or Shakur getting hiked the ball, and Travis Hunter in the end zone, but you don't know what happened between those, right? But we'll see if this works. But if you have a movie, then what you can see is Shakur drops back, he hits Travis in the flat gets one guy to miss, second guy to miss, goes down the right sideline, touchdown. For the aficionados in the audience, you'll notice this was not against Nebraska. Okay. So we can do this in biology, and this is one of the things that we do a lot in BioFrontiers. As Leslie pointed out, we have lots of technologies that we support here, which labs all over campus and even biotech companies in the vicinity use. And so we have really high-end microscopes that are so good, they can actually watch a single molecule move around a cell, which if you think about that, that's just amazing, right? A single molecule moving, right? And then the other innovation that's really important is we can make proteins turn different colors. We can attach to them something that's green, and the tau you're going to see in this movie is going to be green, or we can attach something that's red, and we can make multiple different colors. All right, so here's what a movie looks like. So these are cells. They're human cells in a dish, and they're expressing tau, and we're going to add a seed into them. And what you're going to see happen is you see this cell here, that's that red protein we've been talking about. It's in the nucleus. Look, it popped out into the cytoplasm, and look, that's where tau grew, right? I'll just do this one more time because it's kind of amazing. If I can figure out how to do this. Okay, you can see I'm a bit of a Luddite. But, anyway, so Okay, so what this has told us, wait, why is there another? It's identified a place where proteins grow, and I don't know what. Okay, so that identified a structure in the cell. These tau aggregates just don't grow randomly. They grow in association with this particular structure. We call it a cytoplasmic speckle. And now we've dug into this more. We've identified the molecules in there which interact with tau and make tau form fibers faster. We've showed that if you put that into a mouse, those mice get diseased faster and worse. And now we're trying to understand this in greater detail. And this is work done by some grad students in the lab, Fred and James, and also Kat and Carolyn, who are longtime researchers in my lab. All right. I'm going to pause here and take a breath. So I do this for a couple of reasons. One, I've covered a lot of ground. And this is a chance, if you have a question, I don't know who put this music. Is that you? How do I can't even, anyway, if there's a burning question, I can take a couple of questions here as a transition before we go on. Yes? Yeah, so the question is the immune system involved? And the answer is yes, to a huge degree. So a lot of things that happen in our brain, like the repetitive traumatic brain injury, that's probably triggering what we call neuroinflammation, and that helps drive these diseases. When viruses get in your brain, triggers inflammation and drives these diseases. There's a nasty feedback loop here where if you start making these aggregates and have cells dying, they trigger inflammation, which triggers more. So neuroinflammation is a huge part of this, but I haven't gone into detail today just because I can't cover everything. But yes. Okay. Yeah, one more. Yeah.
Agents that not prevent, say, Parkinson's, but slow it down.
Yeah. Yeah. So it's Howard, right? Yeah. So Howard asked, okay, and so in Parkinson's, we treat Parkinson's with a drug called L-DOPA. How does that work? So what happens in Parkinson's is the first neurons that die in Parkinson's are neurons which make L-DOPA, okay? They use dopamine for their neurotransmitter. And so, so what you're doing when you give people L-DOPA is a molecule, it's a precursor to a neurotransmitter. So you're giving people a drug which allows them to make more dopamine, which allows their brain to work more normally. And so that works for a while, but eventually as you lose more and more neurons, it eventually stops working, right? So that's what L-DOPA does. Well, yeah, and it helps the population to function more for a while, yeah. Okay, one more and then I have to go on. Yeah. I would say there are specific proteins. If every misfolded protein caused disease, we would have 25,000 neurodegenerative diseases. And so it's only some that when they misfold in a way that can trigger their buddy to do the same thing. And those are the ones which turn up as being disease-causing. So it's very specific. And if you want, we can talk at the end about why certain proteins have that property. Okay, so now I want to go on and I want to talk a little bit about why neurodegenerative diseases go up with age and then what we can do about this. And this is the prevalence of Alzheimer's, but it's not just Alzheimer's. Every neurodegenerative disease looks very similar to this. And Leslie, you'll notice that it's a little bit worse for women than for men. Leslie often likes to point out sex differences in her talks. And I should say, but for ALS, it's reversed. It's worse for men than for women. So, okay. So we don't really understand this, but I want to give you a framework of how to think about this. And the way that I think about this has to do with what are called clearance mechanisms. So our cells in our brain have ways to get rid of these kinds of toxic proteins. So we have systems inside of our cell which degrade those protein aggregates, those inappropriate tau molecules. And you can think of this like a cellular garbage disposal if you want, okay? So that clears up your brain. It's not just inside our cells. We also have a system outside of our cells in the brain called the glymphatic system, which flushes spinal fluid through your brain when you sleep, and it washes these kinds of protein aggregates out of the ones that are outside the neurons, out of the brain, eventually into the blood for clearance, okay? You can think of this like your neighbor, hosing his driveway off to get the leaves away, right? Very analogous to that. So what happens then is, in part, is both these systems slow down as you get older. So our ability to clear stuff gets worse as we age. And so if you're young, you can actually have some aggregates. And there's cases where aggregates have been put into kids by mistake, but they get cleared and they don't cause disease for a long time. But as you get older, part of the problem is we don't clear things as well. And if you don't clear things as well, then not only can they build up, but they can also keep spreading. The analogy I often use here is the bucket analogy. So, if you're young, you are producing some of these seeds, but they're being cleared quickly, so you have a low disease burden and you're okay. But as you get older, if your clearance goes down, then you get more disease burden. And these guys can actually then start replicating because they're prions, and they can make more of themselves, and then bad things happen. I wanted to show this simple analogy because, as some asked here and here, there are other things which feed into neurodegenerative disease, and you can put them all in this same kind of format. For example, there are mutations which cause disease in tau itself. And what we know that those mutations do is they make more seeds, and they make those seeds grow faster, and people with those mutations get disease earlier than the rest of us, maybe in their 50s. Similarly, you can think about this for chronic head trauma, because chronic head trauma drives inflammation. We don't really know what inflammation does to this process in the brain, but I would speculate it might affect more seeds, and it might also make, I think probably this one, more seed replication faster. So, but there are lots of different things which impinge upon this, but they can all kind of fit into this general kind of model. All right, so we don't really understand this, but I think clearance is part of the problem. And this is why young people don't get disease and older people do. Okay, I'm gonna skip this because I'm running out of time. All right, what can we do about neurodegeneration? That's what you're all asking, thinking, right? Like, okay, this is really bad, Roy. What can we do, right? So, the bad news is we can't do anything. Well, the first disease-modifying treatments for Alzheimer's were approved a few years ago. These are incredibly controversial, right? So what these therapies do is they target A-beta plaques, those first things that happen, and they get rid of those plaques, and I'll show you how they do it in a second. So those plaques can go away. You get a small decrease in the rate of cognitive decline. So instead of going down at this rate, you go down a little bit slower, right? And that's a good thing, but there are substantial adverse effects, including brain bleeds. And that happens in about 25% of the patients. So you lose your cognition a little slower, but you also have the risk of serious brain bleeds. What these drugs do is they're antibodies that bind to these A-beta plaques and cause them to be engulfed and eaten by the cells called microglia in your brain. These are kind of like cleanup cells in your brain. So they actually work quite well. But there's a problem, right? If you go back to that first thing I talked about, where A-beta happens way before the disease. So we think that about 20 to 30 years before you have symptoms, you start building up A-beta plaques. And then about 10 years before symptoms, A-beta triggers tau to start forming these aberrant structures and to spread. And then about 10 years after that, you start getting symptoms. So that's when you're treating people, right? You don't treat people over here. You treat them when they walk into the clinic because they're having problems. And so we can't really treat people at that stage of disease with A-beta antibodies. What we need to do is treat tau. And so this is kind of where there's an immense amount of work now. I went to the clinical trial database where you can see what clinical trials are proposed. They're not all happening. There's 171 possible trials proposed for tau, and these do a lot of different things, right? There's some that reduce the amount of tau protein. There's some that try to enhance these clearance mechanisms, right? There's some that try to block seeds getting out of neurons or preventing the toxicity. There's some that sequester, just like we had antibodies that could grab onto the A-beta. If these are outside cells, we can grab onto them with antibodies and make them go away. So every possible idea you could think of here, there are biotech companies trying to develop it and push this into trials. Trials are really hard for these diseases because they're slow, progressive diseases, so trials take years, and they're very expensive because of that, so it's really hard to do a trial. 3 of these I pay attention to, and I want to mention why I pay attention to them. One is a drug called an ASO, antisense oligo for the aficionados. This targets tau, and it reduces the amount of tau protein being made by about 50%, and that seems to, people who are treated with this, it's in clinical trials, their tau aggregates clear up a little bit. Not go away, but they clear up some. And so this is one that I think has great potential. Will that reduction be sufficient to change disease? We all think in the field that if we got rid of these tau aggregates, people might get better, but nobody's done that, so we don't really know, right? So it's important to see if that's really true. So this is one I think is, this is a trial being done by a company called Ionis and Biogen. The other one that I pay attention to, just because there's 41 of these on the clinical trial registry, is using antibodies to sequester tau. I think the big issue here is free tau the main mechanism by which tau gets from one cell to the other? We actually know that these seeds can be packaged in little vesicles that get out of this guy, and they float across here, and because they're in a vesicle, they're protected from these antibodies getting to them, and they get into the next neuron. So I think this one, I'm pessimistic will work, but I'd be happy to be proven wrong because I'm getting older. All right, then there's the science fiction idea, all right? So this is the idea, can we treat neurodegenerative diseases with oscillations of 40 hertz? And this goes back to the biology in all of our brains. We have these oscillations that go on all the time, particularly when we're asleep, between 30 and 80 hertz. And just to remind everybody, hertz means once per second. Okay, so 30 hertz is happening 30 times a second. And it's thought that these coordinate different regions of the brain, and it's been observed that in people, in mice, and maybe people who have Alzheimer's disease, that these are gone. And so this woman, Li Shui Sai, at the Pico Institute at MIT, had this idea, which sounds like science fiction, is maybe if we put these gamma oscillations back, we could fix people's brains. Okay, I mean, this is kind of nuts, right? So what did they do? So they take, you can entrain these by sound, light, or tactile stimulation, right? So you can get your brain having these rhythms by external stimulation. And what this sounds like, this is the 40 hertz thing that they play, right? So, okay, so they did a clinical trial, it's well tolerated. The clinical trial was for patients to do this for an hour before they went to bed. And also, they have a synchronized light that flashes at the same time. So they're entraining the rhythms both through their eyes and through their ears, which fits more of their brain, right? And in animal models, it helps get rid of these protein aggregates. And the data now looks like what this does is, remember that hosing off system that helps hose off your brain when you're sleeping? It helps that to work better. And as a consequence of that, this also helps people who have insomnia sleep better, is what they've seen in some of the trials. So, you know, the initial trials show some promise, but they're small numbers of patients and not that long a time. So this is going on now, bigger trials, longer times, and this is kind of a wild card in this area. All right, so those are 3 therapies to keep your eye on. In the meantime, pay attention to your lifestyle if you're worried about these things. There are lots of things. There's a big report just recently from The Lancet, one of the leading medical journals, and they estimate that 50% of neurodegenerative diseases and dementia could be reduced and eliminated by lifestyle choices. So things that increase your risk: smoking, excessive alcohol consumption, high blood pressure that's untreated. So if you have high blood pressure, you treat it, it's okay. Diabetes, obesity, depression. Pay attention to this one: untreated vision or hearing loss. You know, a lot of us think, I don't really need to hear. I don't really want to hear, get grumped out as much. So, but if you don't treat this, you're not stimulating your brain correctly, and then that's causing problems in your function: air pollution, traumatic brain injuries. So, you know, the good news is the kinds of things that you would do to protect yourself from neurodegenerative diseases are the same kinds of things you would do to keep yourself healthy, keep your cardiac health good, maintain robust intellectual activity, keep robust social interactions. Exercise is one of the biggest effects, and eat a healthy diet. All right, in the last few minutes, I just want to tell you about what we do in my lab. You know, as we learn more about how this process works, we're always asking, can we take advantage of that to develop new approaches? And so I'm going to tell you about a few experiments done by Megan, a postdoc in the lab, and we did this work with Charles Hofer, who's a professor here in the Integrated Physiology Department, and Vanessa is a researcher that helps out. And, you know, I lived through COVID, like all the rest of you did as well. I don't think we have anybody under 4 here. So I think about this like a viral infection. You know, that viruses spread between individuals, just like these prion spreads between neurons. And what we learned from viruses, if we vaccinate people, we can protect them and there's no spread of disease. So maybe we could vaccinate neurons and protect them from disease. Now, how do you do that? You know, and I use vaccinate in quotes because we're not going to make an antibody or traditional vaccination. But what Megan did is design proteins which can bind to tau. They have a little part, what we call a targeting peptide, which makes them interact with that tau aggregate, and then it drags in a cargo along. And that cargo, so this is what it looks like. If we just have the cargo, it's off by itself away from tau. But when we put the targeting peptide, you can see the cargo and tau now overlap. And what's really important about this is our best cargo then reduces tau by 90%, okay? So we can prevent, this is all, you know, in a dish, but we can prevent disease by 90%. That's pretty good. But of course, that's in a dish, so is this true in a brain? And so this is how we use different systems. So now we go to a mouse, and we take a mouse which is genetically engineered to have these diseases. We deliver this protein through using a virus, which we can add to their brain and expresses in many cells. And these are the same kind of viruses which are being used in gene therapy treatments in people today. And what we see is in the mouse, we can actually we can protect the mouse by about 75%. So that's pretty exciting, but I should say, a lot of people have cured these diseases in mice. You know, mice are not people. So now what we do in the labs, we try to improve this. Can we deliver later? And can we understand the mechanism? Or maybe even develop a drug to do the same thing? Because if I was to query you, how many of you would be willing to let me stick a needle in your brain to inject virus or take a pill? I think there'd be a preference for the pill. So, all right. So I just want to wrap up then. You know, I've tried to cover 3 areas. You know, what neurodegenerative diseases are. If you forget everything else, remember this aspect of prion spread, because I think that's fundamental to all these diseases, and it affects how we have to think about intervening. And then there's different lifestyles, and you know, we don't have good therapies today, but they're coming, I think. So I'm going to wrap up by speculation. Those of you who know me know that I often get criticized for too much speculation in my papers, but you know, I think that's where you have to think, right? If I have to guess, I would say in the next 5 to 10 years, we're going to have therapies, maybe viral, maybe drugs, that will block these neurodegenerative diseases' progression, okay? I think we're getting there. But the timing is going to be difficult. Because to really be effective, we're going to have to treat before you have those obvious symptoms. Because once you have symptoms, you have a lot of dead neurons, and you can't bring neurons back, at least with today's technology. So this creates some social issues. How do you treat someone before they have symptoms? So I could give you all blood tests, and people will develop good blood tests to do this, and I could say, oh, look, you're at very high risk of Alzheimer's. You need to start taking this drug. Oh, and by the way, this drug has really bad side effects, so you have to think about it, right? So are you going to be willing to take a drug when you don't have any symptoms yet, right? And I think there's also going to be a social issue. Our insurance companies are going to be willing to cover that drug because it's going to be expensive if you don't have any symptoms, right? You just have a high risk, right? So I think there's going to be some social issues here about how these diseases get treated. And then the wild card is this gamma stimulation, because if that works, we're all just going to buy these goggles with synchronized headphones and be doing this instead of watching TV. You're going to be going, So anyway, all right. So I'm going to stop there, and I'll be happy to take any questions. Thank you.